lapel.
Learning her principles of biochemistry. Chapter 1, The Foundations of Biochemistry. Physical Foundations, Part 2. Energy coupling links reactions in biology. So, what exactly is energy coupling? Well, in simple terms, it's the process of using energy from one reaction to drive another reaction. Just like how a domino effect sets off a chain reaction, energy coupling allows living cells to carry out essential processes. Let's break it down. In living organisms, cells need to carry out both energy-releasing, exergonic, and energy-consuming, endergonic, reactions. But here's the catch: endergonic reactions, which require an input of energy, often need a little push to get going. This is where ATP, or adenosine triphosphate, comes into play. ATP is like a cellular battery, storing and transferring energy within the cell. When ATP is broken down into ADP and inorganic phosphate, it releases a burst of energy that can be used to power endergonic reactions. Now, remember the concept of free energy? It's like the currency of the cell, and ATP is the high energy form, while ADP is the lower energy form. The conversion between ATP and ADP is a prime example of energy coupling in action. But how do cells ensure that these reactions happen at just the right time and in the right amounts? That's where enzymes come in. Enzymes are like the master orchestrators, speeding up chemical reactions and ensuring that energy is harnessed and utilized efficiently. In the grand scheme of things, all these reactions are part of a complex web known as metabolism. Metabolism involves a series of interconnected pathways that regulate the synthesis and breakdown of molecules, all while maintaining a delicate balance. Keck and delta G degree are measures of a reaction's tendency to proceed spontaneously. What are Keck and delta G degree? Well, Keck is the equilibrium constant, and it tells us the ratio of products to reactants at equilibrium for a chemical reaction. It gives us an idea of how far the reaction has proceeded towards products or reactants. On the other hand, delta G degree is the standard free energy change, and it indicates whether a reaction is spontaneous or not under standard conditions. Now, let's break it down a bit. When a reaction reaches equilibrium, the Keck value tells us whether the reaction favors the formation of products or the starting materials. If Keck is greater than 1, it means the products are favored at equilibrium, and if it's less than 1, the reactants are favored. As for delta G degree, it tells us if a reaction is spontaneous or not. If delta G degree is negative, the reaction is spontaneous, meaning it will proceed forward without any external influence. But if delta G degree is positive, the reaction is non-spontaneous and won't proceed on its own. So, in summary, Keck gives us an idea of the position of equilibrium, while delta G degree tells us about the spontaneity of the reaction. Enzymes promote sequences of chemical reactions. First things first, let's talk about enzymes. Enzymes are like the superheroes of the biochemical world. They are specialized proteins that act as catalysts, which means they speed up chemical reactions without being consumed in the process. Now, imagine a series of chemical reactions taking place inside a living cell. These reactions are like a well-choreographed dance, each step leading to the next in a precise sequence. But here's the catch: many of these reactions wouldn't happen at a useful rate without the presence of enzymes. You see, all chemical reactions involve an energy barrier called the activation energy. It's like a hurdle that the reactants need to overcome to transform into products. This is where enzymes come into play. They lower the activation energy, making it easier for the reactions to occur, kind of like greasing the wheels of a machine to make it run more smoothly. Enzymes achieve this by binding to the reactant molecules, forming what we call an enzyme-substrate complex. This binding process stabilizes the transition state of the reaction, making it easier for the chemical bonds to break and reform, ultimately leading to the formation of products. So, let's break it down. Enzymes help to lower the activation energy, making the downhill journey from reactants to products much more accessible. This ultimately speeds up the reaction, allowing the cell to carry out essential processes efficiently. But it doesn't stop there. Enzymes also play a crucial role in metabolic pathways, which are like intricate biochemical roadmaps within the cell. These pathways involve a series of consecutive reactions, each one facilitated by a specific enzyme. It's like a relay race where each runner, or enzyme, passes the baton, or substrate, to the next, ensuring that the process continues smoothly. Now, let's talk about the genetic foundations. The instructions for building and maintaining living organisms are encoded in the DNA. Enzymes are responsible for transcribing and translating these genetic instructions into functional proteins, each with a unique three-dimensional structure. Finally, let's touch on the evolutionary aspect. Over time, genetic mutations occur, leading to changes in hereditary instructions. While some mutations may be harmful, others provide a survival advantage in certain environments, driving the process of natural selection and contributing to the diversity of life on Earth. Metabolism is regulated to achieve balance and economy. Metabolism is like the bustling activity of a city, with thousands of different processes happening simultaneously. Carbohydrates, fats, proteins, and nucleic acids are being synthesized and broken down, all in precise proportions required by the cell. Consider the pathway in a cell that leads to the synthesis of the amino acid isoleucine. It involves a series of steps catalyzed by different enzymes, and it's regulated to ensure the right amount of isoleucine is produced based on the cell's current requirements. Metabolism is not just a linear process. It's more like a web of interconnected and interdependent pathways. A change in the concentration of any one metabolite can set off a ripple effect, influencing the flow of materials through other pathways. Now, let's talk about free energy change, which is a key concept in understanding cellular metabolism. When a chemical reaction occurs, it can either release free energy, known as exergonic, or require an input of energy, known as endergonic. ATP, the energy currency of the cell, plays a crucial role in driving many cellular processes. Its breakdown releases a large amount of energy, which is used to drive endergonic reactions in the cell. Entropy, or the measure of randomness or disorder in a system, is another important factor in metabolism. Living organisms exist in a dynamic steady state, far from equilibrium, and require a constant investment of energy to maintain this state. Cells also regulate the synthesis of their own catalysts, the enzymes, in response to increased or diminished need for a metabolic product. This adds another layer of control to the overall regulation of cellular metabolism. So, there you have it. The intricate dance of metabolism, where thousands of interconnected pathways work together to maintain balance and economy in living organisms. It's truly a marvel of nature. Thanks for tuning in. If you enjoyed this video, don't forget to like, share, and subscribe for more fascinating insights into the world of biochemistry. See you next time.
Pharma Pal.